So based on the docking studies, what we can see here is we can see the rigid versus flexible docking here. So if you see what is a rigid versus flexible docking is, the rigid body docking, that is no modification in the bond angles, lengths and torsion angles of the components will be there. Flexible docking means into the conformational changes, that is based on the conformational changes they can have the flexible docking, that is in which angle they have to take, that they can decide. So this is how the process actually is happening. So you must have seen this in your childhood, that there will be some puzzle. Okay, so in that puzzle, if you fit it properly, they can fix like this. So in the same manner, this is like a ligand and this is like a receptor. So they will have a specific position in which they can interlock themselves. So the protein docking is a computational determination of protein complex structures from the individual protein structures here. So this is given by Smith and Sternberg in 2002. So the interaction, what happens? The interaction leads to the ligand binds to the active site of the protein. The binding leads to the conformational changes in protein, and the conformational changes thermodynamically becomes most stable, and the lowest Gibbs energy is being determined here. So in the same manner, when as per the Smith and Sternberg of 2002, they say that the protein-protein interaction thermodynamics, how it looks like is, it is by delta G, that is minus RT ln by RL by R and L. So here you can see that there are many different protein structures are there. When they are binding, okay, when they are having some interactions between these pink colors and green colors ones, they are interlocking themselves. Once the binding free energy actually decreases, because this is free energy is required, so once the energy actually decreases, they can again go back to the normal state. So that is why it is vice versa format which is present here. So why docking is important? It is very much essential or extremely relevant in cellular biology, where function is accomplished by proteins interacting with themselves and with other molecular components. So it is the key to the rational drug design, because here what is happening is the result of the docking can be used to find the inhibitors for the specific target proteins and thus to design the new drugs. So basic idea is, whenever we want to design something new, okay, so new drugs, when they are designing, this docking actually reduces our time. The basic idea is to reduce our time and effort so that we can release our product at the earliest and faster in the market, so that the profitability of this particular drug or the product, pharma product, is very high. So what is the basic requirements for docking? So basic requirements for this is to know the structure, protein structure of interest. We should know about the X-ray crystallographic study, and the NMR spectroscopy has to be conducted. So X-ray diffraction, as explained by the Hubbard in 2006, they say that the spacing of the atoms in the crystal lattice can be determined by measuring the locations and the intensities of the spots produced on the photographic film. So the X-ray analysis of the sodium chloride crystals, because this is a very commonest method by which they have understood that there is a particular channel and the structure of this particular chemical is actually providing us the detail, that is how the lattice is basically. So the X-ray analysis of the sodium chloride crystal shows that the sodium and chloride ions are arranged in a simple cubic lattice. So instead of having something different, it is having a cubical structure in the lattice. So the wavelength range, if you see, that is the 0.7 to 1.5 Armstrong, that is 0.07 to 0.15 nanometers. So that is the wavelength. So this is how the X-ray diffraction actually takes place. The X-ray source is there. X-ray beam falls on the crystals. That crystal ranges, different part is being, that is the diffracted beams are there. So that is being determined on the X-ray film or by other sources. The detector is actually capturing it. So if you see, if we are having the crystals, and from that the X-ray which is there, that the differential diffraction pattern actually follows. So there are different phases in which we will get it. So the electron density map is then made. See, for example, for certain chemicals it will be very high, certain absorption will be very low. So based on that, the structure will be determined, which angle it is present, in which side this particular chemical is present, or an amino acid is present, and then based on that, by the computational model we can make the atomic model and we can present it. So as per the James paper of 1998, the modern NMR techniques are being used to determine the structures of the larger molecules. So the NMR, nuclear magnetic resonance, so it is based on the nuclear spin angular momentum. So here what we are doing is only certain atoms, including the 1H, 13C, 15N, 19F and 31P, possesses the number of nuclear spin that gives rise to an NMR signals. So you can see here how each lipoprotein in the subclass broadcast a unique NMR sound. So H1, H2, H3, H4, H5, L1, L2, L3, ideal, V1, V2, V3, V4, V5, V6. So like this, it has been determined. So that is the simultaneous linking of the plasma lipoproteins produced a recorded signaling. So how it is being present? So you can see the size of the different groups or the signals derived from the computer analysis give the subclass of the concentration. This is the recorded plasma signal, how it looks like, based on which you can determine the different things like HDL, LDL and VDL, VLDL and all these things. So why is this difficult? Basically, why is many people, they say that docking is very difficult. The main reason is both the molecules are flexible and may alter each other's structure as they interact. So when they are interacting, there are hundreds to thousands of degree of freedom is there. So thousands are there. So they have to search for the particular location in which they can bind and their active site is there. So that part actually takes a lots and lots of time. That is the main issue in this. So as per the Fernandez et al. Paper of 2005, based on the optimal docking areas, a new method for predicting the protein-protein interaction sites, it shows that in this paper, there are some techniques are there, which is being explained here regarding the protein-protein docking here. So the surface representation, if you see, there are two types are there, surface representation and surface matching. So surface representation means what? That effectively represents the docking surface and the identities, the region of interest, that is cavities and protrusions, cannoli surfaces, the clustered surfaces, clustered spheres or alpha spheres. So all these things are alpha shapes. These are the types of the representation of the surface representation. The surface matching, if you see, the matches surface to optimize a binding zone. So that is the important part here. So if you see the surface representation as per the Fernandez et al. Of 2005 paper, it shows that each atomic sphere is given the van der Waals forces. Okay, van der Waals radius of the atom, you can say. So the rolling a probe sphere over the van der Waals surface leads to the cannoli surface. This is actually called as the cannoli surface. Cannoli means you can say that there are something which is being joined and looks like everything is having web almost. Okay, so that is what is the cannoli surface looks like. This is the clustered sphere. Clustered sphere means use the clustered sphere to identify the cavities on the receptor and the protrusion on the ligand, and the regions where the cavities on the receptor or the protrusion on the ligand which is present. So you can see that how the clustered spheres are being formed and how the overlapping is taking place, whether any differences are there, all these things is discussed here. Then alpha shapes. So you can say alpha shapes means what? Alpha shapes means almost, you know, that is almost, you can see the shape. If you take like this is forming an alpha. Okay, so this is how the alpha shape is there. So edges wise. So in the 2D, an edge between the 2 points is alpha exposed. So 2 points means almost you can say this and this are there. So in this, if you see this particular part, there the alpha exposure can take place. If there exists a circular radius alpha such that 2 points on the line of the surface of the circle and the circle contains no other points from the point set, so then it is forming the alpha circle. So you can see here it is actually passing through 2 points, one here, one here, so it is passing through that point. So when it is pointing like this, lie on the surface of the circle, so lie on the surface of the circle, the circle contains no other points from the point set. So this is how the shapes actually looks like example wise. So if it is a plane like this, if you see after the processing, it will look something like crumbled paper. So almost if you are crushing the paper and throwing somewhere, so it will look something like that. So this is the 3D representation of the protein binding site. You can see that how much angle is there, distance is there, torsion is there, everything you can see in this. And you can see that what joint it is present, how it is present, so everything can be analyzed here. So surface matching. So surface matching, if you see as per the Fernandez et al. Paper of 2005, it says that find the transformation, that is a rotation plus translation that will maximize the number of matching surface proteins from the receptor and the ligand here. So there are two things are there. That is the first condition is find the best fit of the receptor and the ligand. And the second is use energy calculations to refine the docking. So here what is happening is you have to find the minimum, that is minimum energy which fits properly there. That is the optimum one here. So as per the paper of Gray et al. In 2003 regarding the protein-protein docking prediction for the Capri experiment. So Capri means critical assessment of prediction of interactions. So in this, it is launched to assess and compare the current docking algorithms and to stimulate further development in the field. So that is the main important aspects of the Capris. So if you see the Capri challenge of 2002, the 7 Capri docking targets are there. So you can see that what are the different receptors and ligands which are present here. So I will take just 2 examples here. That is HPR kinase is HPR, which is the ligand here. And you can see the alpha amylase, that is the AMD10, which is from the Cambiola lab. So that is what is actually the receptor and the ligand particularly. So as per the paper of Gray et al. Of 2003, another paper is there, that is the protein-protein docking with simulations, simultaneous optimization of the rigid body displacement and the side chain conformation. So in this paper, what they are discussing is mainly they are discussing about the docking only. So they are saying that docking has 5 steps here. So step number 1, that is the start the crystal coordinates of the target receptor. Step number 2, generate the molecular surface of the receptor. Step number 3, that is the generate the spheres to fill the active site of the receptor. That is the spheres become potential locations for the ligand atoms. And step number 4 is matching. Matching means spheres centers are then matched to the ligand atoms to determine the possible orientations for the ligand. And step number 5 is scoring. That is find the top scoring orientations here. So in this docking protocol, if you see, the basic thing is we have to find the random start position. So random start position means once the structure is there, in that structure, that is you have to search the particular position where it can bind. So that is your active site searching. Okay. So after searching that, you have to search for the low resolution Monte Carlo search. You have to do it. So low resolution means how the energy is and other things. So we will see that in detail now. So high resolution refinement, you have to do it. Then you have to make it to almost like 10 to the power of 5x. You have to make it. Then clustering and then prediction. So all these things are taken up. And along with that, there is a low resolution decoy is also present. So low resolution decoy means like a copy. You can say like a copy which is being formed. So the repack the side chains, then rigid body minimization, then Monte Carlo, if it is accepted, then it is going to the next step like 50x, and then rigid body move again takes place. Otherwise, it will be again filter and then rejected if it is not, okay, or going for the clustering. So in this docking protocol, the first thing is the random start position. So random start position means a creation of the decoy begins with the random orientation of each partner and a translation of one partner along with the line of protein centers to create a glancing contact between the protein. So you can see that a random orientation is there. Random orientation means there will be not be any particular structure. It will be like a random structure. And it is like a decoy. Decoy means like a dupe. You can say like a, to make it alert or make it a copy. So many copies are being formed. Then based on that, what they do is they will find the location in which the particular protein can bind. Okay, so that they do it. So then they go for the there is a low resolution Monte Carlo search. That is one partner is translated and rotated around the surface of the other. And the score is based on the correctness of each decoy and the residue interaction terms here. And the high resolution refinement, if you see, that is like side chains are added. So if you are having any polypeptide chain, in that polypeptide chain, they have made some structures. In that, they will add the side chains, that is H or other things. So side chains are added to the protein backbone to changing the energy surface. And a filter is employed to detect the inferior decoys. Inferior means which is having very low resolution or something like that. They will find that. And if it is very low, they will reject them without further refinement. Then comes the clustering and prediction. So clustering means the search, the procedure is repeated to create the approximate 10 to the power of 5 decoys per target. Then the best, that is the 200 best scoring decoys are then clustered. Then the clusters with the most members are selected as the final predictions and ranked according to the cluster sizes then. So what is scoring function? So scoring function, if you see, to evaluate the interactions to discriminate the observed mode from others, then binding affinity of the complex to be worked out, and then energetically favorable complexes to be predicted, then large number of degree of freedoms to be considered. So all these things are taken care. This is all about the scoring part. Scoring part means simply to say they actually provide us the data, and they find out that which is the correct binding site, and based on that, the angle and everything, they will give a particular score that in that order they can bind it. So I hope that in this session you have got an idea about the protein docking. So in the next session, we will talk about the protein ligand docking in detail. So thank you and have a good day. See you in the next session. Bye.